ahead.
Yeah.
Take your time.
So, welcome everyone here. Once again, we are back with a good and enriching talk on cancer biology. And we have with us Dr. Jaswandi Dandekar. She completed her BSc in 2016 and her MSc around her MSc in 2017, and then she joined as a research assistant, project assistant, and she completed her PhD in from IIT Bombay in Professor PJ Bhatt lab in specializing in cancer biology, and then she joined Marwadi University as an assistant professor. And after that, in 2022, she joined MS University as an assistant professor at microbiology department. So she's right now there, and she's working with everyone there. So that was a short introduction of Jaswandi Dandekar, ma'am, and maybe Dhairya, you could continue from here.
Uh, Thank you, Ajinkya, for giving a very good, warm introduction of Dr. Jaswandi for today's event. I hope I am audible.
Yes, go
Ah,
ahead.
yeah, yes.
Okay, great. So, Ajinkya is my friend and he is my co-host today for the event. So, Ajinkya is doing his PhD at a microbiology department, and we have been known each other since last 6 years, I guess, 6 or 7 years.
Yes.
So when I joined my bachelor's, master's, and yeah, uh, and right now I'm doing my PhD at University of Freiburg in Germany.
Oh, great.
Not on cancer pathways, but similar pathways which are related to cell death, apoptosis,
Mm.
and cancer pathways like P53, PUMA, BCL2 family.
Correct.
So basically, yeah. So we would like to, talk more about your experience today rather than a monotonous webinar, because like
Correct.
everyone is young here and
Right.
most of them are not much interested in uh, talk just listening every time, and that to which they don't get exactly.
Right.
Rather, maybe we can today talk about you and how was your journey. So I would love to ask you this that how was your experience in your bachelor's and master's, and like when did you had a trigger that okay, I want to pursue my PhD, or I want to go to research or science, that I can contribute something to the society.
Okay. So yeah, thank you, Dharya. And before starting, I would like to thank, the entire team of the SciComm Synapse and obviously Ajinkya for giving me this opportunity to interact with the young minds. And to answer your question, Dharya, so I did my graduation in microbiology from Nasik, the college
Mm-hmm.
which is affiliated to University of Pune. And
Mm-hmm.
obviously, as you know, everybody goes in the middle class family that you want to either
Mm-hmm.
go for doctor or you want to go for engineering. But yeah,
Mm-hmm.
somehow because of so many obstacles, I could not get into either of the branches. And I was not very keen on repeating the entrance exams one more time, dropping year or something like that. I was like nothing doing, whatever comes my way, I'll go ahead with that. But
Very
yeah, I
true.
had that instinct somewhere, I should have doctor in front of my name. But I thought,
Mm.
fine, destiny had something else for me. So I started with BSc microbiology back then. And at that time, it was obviously a very budding field in the science. And so I wanted to pursue that. And obviously, then I developed interest into it. And then, destiny really had something good for me in the pocket. So I ended up doing my master's from MS University, Baroda itself.
Mhm.
So at that time, obviously, I was not aware of this university because I hail from Maharashtra. So Gujarat
Mhm.
is just
Mhm.
a state where Garba and Dhokla is famous. That's all I knew. So yeah, but my friends, they were somewhere from Gujarat. So they told, no, Jaswandi, we should at least appear for the entrance. Let's see what happens. And yes,
Mhm.
thankfully, I got into masters in microbiology at MS University. And at that time also, it was, I had this what I can call it as that future plans as such, that I
Mm-hmm.
will
Mm-hmm.
complete my masters, then I will do MBA, and I will join some industry and have some corporate job, so and so
I
forth.
see.
Yeah, but but thankfully MSU happened to me, and most of the I can see some of my students here, they know this story, that thankfully
Mm-hmm.
MSU happened to me. And the kind of training that we get at MS University, specifically our
Mm-hmm.
department of Microbiology and Biotechnology Center, so
Mm-hmm.
during that journey of my dissertation of almost 2 years there, I
Mm-hmm.
got that, okay, no, this is very interesting. We are trying to deal with living systems, we are trying to manipulate them, obviously in
Mm-hmm.
a good way, so that we can find some basics aspect of life as such. So I guess that was
Mm-hmm.
the trigger point for me to deviate from the MBA and corporate job
Mm-hmm.
and land into the
Mm-hmm.
research field. So yeah, then I followed the
I
normal
see.
part, that entrance exams, then appear for JRF or something like that. I
I
cleared
see.
CSIR and I got into IIT Bombay. And then
Mm-hmm.
obviously that in IIT Bombay, it was very enriching experience.
Mm-hmm.
So,
Mm-hmm.
for that, the all credit goes to my PI, Professor Jaydev Bhatt. He's retired now, but still very enthusiastic for science.
Mm-hmm.
And
Mm-hmm.
one thing he always tells, even today itself, that you
Mm-hmm.
should look at this science as, your religion, not as you're doing something, okay, I want to publish papers, or I want to do this, do that. Yeah, obviously that is a need of time, but of
Mm-hmm.
course you need to look at science from different perspectives. And he
Mm-hmm.
always tells us to try to read between the lines. Anybody can get the message what is written there, but you should develop that habit and try to understand, what the meaning at the global level. So yeah,
Mm-hmm.
working with Bhatt sir was very enriching experience. And he is a wonderful personality, and he has
Mm-hmm.
this, I would say, inclination towards finding complex things, looking just at the phenotypes, right? No high funda
Mm-hmm.
techniques. Obviously
Mm-hmm.
sometimes you might have to go for, let's say, microscopy or facts analysis, so on
Mm-hmm.
and so forth, but
Mm-hmm.
his aim starts with try to look at a phenotype. Because, and that's why, yeast, as I would be talking about it, the eukaryotic
Mm-hmm.
system, it is such a versatile system that,
Mm-hmm.
okay, you just have to have that knowledge that which pathway I want to trigger, what phenotype
Mm-hmm.
I would get, and then you can do wonders into it. So when I'm
Mm-hmm.
discussing my doctoral study, you can see almost 60% of the work is based on the phenotype. And
Mm-hmm.
that is something, very amazing for me at that time.
Mm-hmm. Okay.
Yeah,
Yeah. I mean,
Yes.
I would say science is all observational based. If
correct.
you start
Yeah.
about 1600s or even before that,
Right.
people used to use medicinal plants and for various diseases. I would say even maybe for cancer. We
Perfect.
never
Yes,
know.
yes. Yeah, of
So
course.
maybe 1,000 years back, and this was all due to observational based. That okay, if you apply this leaf or if you apply this root, then this
Correct.
thing happens. Like
Yes,
turmeric
yes,
is one of the
exactly.
best
I mean,
example, I would say.
Right.
there was this plant extract which was coming up for cancer studies. Taxol, I think that's a
Yeah,
plant
yeah.
extract, the stem extract of
Uh-huh,
a plant.
Correct.
uh-huh,
So that was very
uh-huh,
effective. Seeing, yeah, yeah, it was seen very effective, having an anti-cancer property.
uh-huh,
Having
Yeah,
some,
even the betel leaf, if you know, the pan what we eat, that
yes,
uh-huh,
also
yeah,
has been shown to have
yeah,
anti-cancer properties. Yeah, they
yeah,
are
yeah.
exploring,
Even
aspect of that.
yeah, even it has a good digestive property, and that's why it is
Exactly.
said that you should always have pan or betel leaf
Correct, correct.
after your
Right,
lunch or dinner.
right. And
yes,
So
that I can connect to my research topic is that if the digestion is proper, everything is metabolized through oxidative phosphorylation. It
I
rarely
see.
goes to the fermentative pathway, which is,
Hmm.
the key aspect of the cancer cell. So obviously it might have some
And
connection there.
I see, and especially yeast.
Yeah. True.
So yeah, but I was also working with my yeast when I did my thesis, my
Okay.
master's
Huh.
thesis,
Hmm.
and my boss used to say, east or west, east is the best.
True. I totally agree with that.
So yeah, okay. So where did you do your dissertation? And like I would say that especially your thesis, because you told initially you wanted to do a MBA, right? And
Right.
there might be many of them who want to do MBA.
Want
So
to,
of
Correct.
course
yes,
you might
pursue.
have some introduction that how to switch from science to MBA, and then what the specific point? Maybe I would say your master's thesis
Yeah,
could
correct.
be a
Yeah,
turning
that
point
was
for
the turning point,
yourself
of course.
that that led to doing a PhD rather than a 9 to 5 corporate
Corporate.
job. So maybe can you talk a bit more about
Yeah,
your
sure, sure. So
master's
obviously,
thesis?
I would like to say that after MSc, even somebody goes for MBA, that is also an excellent career option. No offense against it, but it's just
Mm-hmm,
that I develop inclination towards science and research, so
mm-hmm.
I pursue that. So as Zaire was asking about my journey for the MSc dissertation, I did my dissertation with Ajinkya's PhD guide, Professor Anuradha
Ah,
Nalekar. Yeah, and at that time she was working with the biocontrol of soft rot causing Irvinia. That time it was called as Irvinia. Now the strain name has been given as Pectobacterium, if I am not wrong.
Pictobacteria. Yeah.
So she was working with that system, and it was the project about finding a biocontrol, not by
mm-hmm,
killing the organism, but by deciphering with the pathogenesis as such. So to
mm-hmm,
tell you in
mm-hmm,
short, the Erwinia has a quorum sensing mechanism for pathogenesis.
mm-hmm,
So all
mm-hmm,
quorum molecules, the AHL molecules, signal molecules basically,
mm-hmm,
if we try to
mm-hmm,
find an organism which can degrade those AHL molecules, obviously that will be a biocontrol, because though
mm-hmm,
the bacterial or the pathogenic population would be more, but they will not be able to communicate with each other, and hence there is no pathogenesis. So that was the basic
mm-hmm,
idea about it. And again,
mm-hmm.
coming back to the same point, that time also the key observation was the phenotype.
I
That again
see.
we use
Yeah.
the Irvinia cells, the Pectobacterium, I would now call it as Pectobacterium, and we
Mm-hmm,
try to look for microorganisms isolated from various soil samples, rhizospheric soil, non-rhizospheric soil, and we try to look for if there is inhibition of the soft rock onto the potato slices as such.
mm-hmm,
And it was very interesting work back then also. So obviously I got few strains which were showing that activity, and thankfully I could, not, I would not say finish a project, there is no end to an any project, but I could come to some concluding point where I got one bacterium, Crysiobacterium, if I'm not wrong, which had that property that it was synthesizing AHL acylase, and that was going to degrade the AHL molecule, so the Pectobacterium pathogenesis would not be there. So thankfully,
mm-hmm,
because of Anuradha madam's guidance and my intermediate, Dr. Vimal Maisuria, both
mm-hmm.
of them, they encouraged me so much into this research, and obviously by God's grace, I mm, got going, with the positive results. So obviously that ignited that spark of finding something new into me.
Mm-hmm.
Yeah.
I see. I see. I see. So I was also MSU product, I would say.
Oh, is it?
I was, yeah, yeah, I'm also a MSU product.
Acha.
I was in the same department where me and Ajinkya completed our bachelor's and
Oh,
masters, that is
CMB.
integrated. Yes.
Correct. Yeah.
So I was like initially from my first day, I would say, when I joined my bachelor's, I was working with Professor Rashidunisa Begum in the field of vitiligo and
Ah,
cell
right.
death, majorly
Correct.
on cell death
Mm-hmm.
and autoimmunity and these kind of things. So from the starting only, I was inclined towards molecular biology and
Right.
you know cell death pathways, that how a cell
Correct.
die, how a cell die, you
Hmm.
know. So and similarly a cell, how a cell proliferate. Like, I
Of
would
course.
say
Yeah.
almost all mechanisms
Hmm.
are same at the point.
Interconnected. Yes. Yes.
Yes, the pathways are completely interconnected. So, I would like to ask you this question now. That how did you change your field when you
That's,
joined Anuradha's ma'am for microbiology,
yes.
doing some microbial stuffs
My
and like that, and then suddenly into the eukaryotic system and working towards the proliferative pathways and cancer pathways. So
Yeah.
what
Okay.
made you shift over there, and maybe can you talk a bit more about your experience when you joined the your experience of CSIR net? I would say I
Hmm.
would I would love to
Oh.
hear that, and uh, what was your tactics to clear it? Because I would not much has changed yet,
Of
so
course,
there are
true.
many young people over here, and also few of them are listening on our YouTube, because
Correct.
we couldn't give entry to everyone.
Right, right.
So yeah, so what would you like to say? Maybe you can elaborate a bit on the condition of CSIR net that everyone is facing right now, and yeah,
True. Yeah,
that
okay.
would
So
be
to
really good.
answer that question, Dhairya, of course, okay, when I cleared my CSR exam, the pattern was quite different. It was subjective plus objective kind of an exam. So it was like you will attempt both the set of questions, objectives as well as subjective. And subjective were, trust me, were very difficult to crack, because it was not just write a short note and blah blah blah. It was like you'll be given some actual problem, scientific problem,
Mm-hmm.
and you will have
Mm-hmm.
to come up with a solution to tackle that problem, something like that. So
Okay.
it would be really brainstorming if you want to answer those questions.
Mm-hmm.
And
Mm-hmm.
on top of that, sometimes somebody has inclination towards analyzing something, but they might not be good in multiple choice question type of
Mm-hmm.
answers, right?
Mm-hmm.
But
Mm-hmm.
the catch there was, you will appear for both the exams,
Mm-hmm.
but when your paper would be assessed, it will be the objective part that will be first assessed, because it was OMR sheet, just it have to be fed to the machine will do the task.
Yeah. Yeah.
And if a student or a candidate is above a threshold level in that objectives, then only
Mm-hmm.
his or her subjective part would be taken into consideration for
Oh,
evaluation, right?
okay.
So yeah. So even if you do great in subjective, and if you fail to do that much in objective, it was of no use. So you had to be on your toes in both the aspects, because obviously if you clear objective, having good score in subjective was also equally important. So
Mm-hmm.
obviously to the students who are the aspiring scientist, I would say, want to clear entrance exams, my
Mm-hmm.
only suggestion to them is, please pay attention to the basic understanding of any scientific phenomenon, any scientific mechanism, rather than going into the glorious part of it. Simply, if I want to tell you, let's say, just now for the sem 1 students, I have taught them restriction endonuclease enzymes. Okay, so if you look from normal perspective, it is like, okay, enzyme will come, cut the DNA, blah blah, thing is done. But you need to understand, okay, in a plasmid there are few restriction sites. If I cut with
Mm-hmm.
one particular enzyme, how the digested product would be. If there are multiple enzymes, what all things can happen. If it is partial digestion, what is going to happen. If it is complete digestion, what all things can happen. So it is important to understand the basic of any concept, scientific concept. Then it is, then on that you can build up the story to memorize
Mm-hmm.
something. Okay, इस enzyme का नाम suppose याद रखना है, then all those are रट्टा मारू things. But for that,
Mm-hmm.
first you need to have the understanding of the topic. Similarly, let's say
Mm-hmm.
cell cycle regulation, if you are studying, just
Mm-hmm.
merely mugging up, okay, CDC 25 cyclin kinase, they do blah blah blah. Okay, it's not going to work. But you need to understand, if that particular cyclin CDK complex is doing something in S transition phase, why it is doing so, why it is that so
Mm-hmm.
important.
Mm-hmm.
If you get a knack of that, and if you get a hang of that, basically, then
Mm-hmm.
your mind will start thinking if, when you are facing any analytical problem.
Yeah.
And I
Mm-hmm.
guess that habit is going to thrive them during their research journey as well, because you both might know by now that doing research is not child's job. It takes your sweat and blood out of you. So unless and until you have, even for understanding a simple experiment from a research article, you need to have the background study done for it.
I see.
So yes, the only key, again, coming to the point, that the only key to crack the competitive exams is, you have to be regularly into it. It's just not that ki 15 din
Mm-hmm.
pehle I will study and I will crack CSR. Not going to happen. Entire year 2 years of masters, and even I would like to add 3 years of bachelors also. So complete
Mm-hmm.
5 years, whatever has been taught to you, and whatever extra knowledge you can gain from,
Mm-hmm.
let's say, social media, from internet, whatever, try grasping
Mm-hmm.
that knowledge, because
Mm-hmm.
you never know which thing might help you anywhere. So I
Mm-hmm.
think that is the key, that it is a continuous process. It cannot just happen overnight. And
Mm-hmm.
to answer Dhairya's second question, that how I switched from bacterial system to the eukaryotic system. So to answer that, yeast is a very unique system in that manner, that it is an eukaryotic system, but the laboratorical handling of that system is very much alike any bacterial system, right? So obviously, and at that time, okay, to tell you even in details, when I applied for different institutes for once I cleared my CSI, I applied for various institutes for the doctoral program. And before that, after my masters, I was working in National Center for Cell Sciences, that is in Pune. I was working
NCCS.
at, yeah, NCCS Pune. I was working as project assistant there, and obviously there the entire work is only onto cancers. They work
Yeah, yeah,
nothing but cancers, right?
yeah.
And
Majority.
yeah, majority of them, rather I would say almost 90% of the faculty is there, they're working on some or the
Mm-hmm,
other cancer type.
Cancer.
mm-hmm,
So
mm-hmm,
obviously I got exposure to cancer field as such there, but
mm-hmm,
somehow, okay, that might sound very childish and very naive, but they
mm-hmm,
work with the mouse models and rat
mm-hmm,
models, and
mm-hmm.
obviously you'll have to kill them and do all the studies before that.
Yeah,
And
yeah.
somehow I was not very comfortable with that. So
Mm-hmm,
killing somebody was a bit a task for me. So when I was
mm-hmm.
doing project there, I had to kill mice twice or thrice, and then I could not eat for next 2 days. So
I
yeah, I
see.
understood that okay, this is not my cup of tea. Obviously
Mm-hmm,
others are doing that, but personally I don't know what went wrong with me at that time, but I was not able to cope up with that.
mm-hmm,
Rest everything
mm-hmm,
I could do fantastically, immunohistochemistry, western blot, RT-PCR, you name it, I was able
mm-hmm,
to do it. But this
mm-hmm.
particular thing, that dissecting a mouse and taking tumor out of it, was a task for me. So I thought, okay, that might not work for me in the long run. And at that time, the not recruitment, the PhD projects at IIT Bombay were also floated. So there,
Mm-hmm.
in that session for intake, Bachelor had floated this project, that the
Mm-hmm.
role of P53, understanding role of P53 in cancer
Oh,
metabolism using yeast
mm-hmm.
as a system. So I thought, okay,
Okay.
I wanted to work with microbial system, I wanted to work with cancer, but if I work with microbial system, no need for dissection, so let's try that.
Hmm.
So I
I see.
applied there. Yeah, in IIT also, even if you clear CSIR gate, any entrance exam, they have 2 rounds of interviews, and after
Mm-hmm.
that you will get selected. So thankfully I got selected, and
Mm-hmm.
thankfully Bhatt also wanted me to join his lab. It goes
I
mutually,
see.
that student will give preference to the faculties, and faculties
Mm-hmm.
also give preference that which student they want to take into their lab. So thankfully
Mm-hmm.
both of us, the that intellect clicked, and we started working together.
I see. I
Yeah,
see.
that's
So, was this a typical IIT Bombay interviews that happen like with there are, I guess, when I was doing my internship under Dr. Sanjeeva, he
Sanjay
works on
Srivastava, yes,
proteomics and he
yes,
works on proteomics. So I
correct.
was in his lab for 2 to 3 months. So at that
Oh,
time,
you
the
were in a very
education.
glamorous lab of IIT Bombay.
No,
Are he
yeah,
is hero of BSB, I must
I
tell
would
you.
I would say yes, that is a correct term, the glamorous
Yeah,
lab. I mean, which the lab is, of course.
correct.
So I have not visited IIT Bombay since last many years,
Okay,
but yeah,
so
I really want
when you were in IIT Bombay, the department was in the old building or the new building?
new building. I
Ah,
guess
okay, fine.
new building. Yeah,
Yes,
because my
correct.
lab was in new building, bioscience and bioengineering department.
Correct. Third floor lab.
Yes, third floor corner one. Correct.
Okay.
So yeah, yeah, so I wanted to ask that how was your interview, experience? Were you like really nervous when you went there? Because when I saw the people, they're like, okay, some of them are sleeping on the couch, some of them are doing this, doing that, and everyone is like nervous, and you see that real tension, I could say. And then that day I could
Anxiety.
actually
Yeah.
imagine that, oh God, getting into IIT Bombay after CSIR net is also quite difficult, I see, with so many
Yeah, of
people.
course. Of course it
So,
is
so can
difficult.
you tell
So
me
yeah,
about your interview a bit?
yeah, okay, sure. So obviously, as I have mentioned earlier, I am a middle class, typical middle class girl. So I go through all this anxiety, nervousness, and to add to that, I would like to mention that when I was doing my bachelor, So bachelors now everybody they try for MSc ki chalo IIT mein mil jaye, jam ki exam dete ye wo. But during my bachelor's I was so confident that I'm not going to get into IIT. So I did not even appear for the exam, right? So that was the confidence level I had that I'm surely not going to get into IIT. So क्यों पैसा बर्बाद करना types of thing, okay? But that was the wrong thing. I think what I did. At least I should have appeared for that. You never know. I might have done masters from IIT Bombay itself. So for these young minds who are here, do not even leave any one stone unturned. Whatever you can do, just go for it. Whether you clear it or no, that is a different issue. But what mistake I did, you please don't repeat that. So yeah, at that time I was like, yeah, IIT अपने लिए नहीं भैया, वो तो बड़े लोगों के लिए होता है, ऐसे वाला था, right? But then, thankfully, the training that we get at MSU, that gave me the confidence. Okay, at least let me try applying for IIT Bombay, right? So obviously I applied for IIT Bombay. I also had applied for NCCS. I had applied for CCMB at Hyderabad and everybody. So now talking about IIT Bombay's interview, obviously it was very nerve wracking, because as I mentioned, you have 2 rounds of interviews. So in the first round, there is a panel of 3 faculties together, and they will get distributed. So 3 faculties in one room, other 3 in other room, and students would be distributed amongst them. Okay, 10 students for panel A, 10 students for panel B, something like that. And the questions what they ask there are all logic based. So again, coming to point what I mentioned earlier, if you have the clear understanding of a topic, then only you will be able to answer those questions. They won't ask you to define what is pathogenesis. No, they're like, you should be knowing that. Let's move ahead that, right? So it goes like that. So once that round is over, they will select few candidates. Every panel of the teachers, they will select few candidates, and now those selected candidates will face the interview, where entire departmental faculty would be sitting. So it's like 10-12 faculties in front of you, and you are only one sitting there, and from everywhere the questions will be bombarded at you, and you have to answer those. So again, if your first thing is, you have to have the knowledge of the subject what you're talking about. And second important thing, obviously I understand it might not be possible. Even today I might not be knowing all the aspects of all the topics whatever are there in the science. So if you don't know anything, do not try to bluff. You just tell them ki I'm sorry, I'm not aware of this topic, so can you ask me something else? That is perfectly fine. But what happens, whoever are sitting in front of you, they're the Maharajas of their clan, so you cannot try to fool them. So even if you give tiniest loophole, they are going to make tremendous use of it. And you will be left nothing in your hand. So better is, be honest to yourself first. Okay, I don't know this, and then spell that out, obviously in a polite manner. And okay, again, that should not be the case that whatever question they ask, you are saying I don't know, I don't know, I don't know. That should not be the case, obviously. So again, the continuous reading of reference books, continuous reading of research articles. I may not say research articles, at least review articles, that is going to give you really a good insight into a particular topic. So yeah, IIT Bombay, interviews are I would say fantastic.
I see. So, so I'm audible, right? I think.
Yes, yes,
Yeah,
yes.
go
Yeah.
ahead.
Okay. Okay. Good. Great. Great. So I see that you had a real motivation. I would say.
Yeah,
And
exactly.
would you
So,
would you now being
True.
a full time professor at MS University or lecturer, would you like to tell the students that are doing their bachelors or masters, whether your students or some other university student, can you focus a bit on reading from PPTs? Because that's what most of
I am
the
totally
people
against
do.
it.
yeah,
I am,
that's
yeah, I am totally against it, because see, I'll tell you. As Ajinkya mentioned, I joined MS University last year. Okay, so
Mhm, mhm,
obviously before that I was working in Marwadi. There I was
mhm,
teaching few topics, so I had
mhm,
PowerPoints ready for it. But now when I
mhm,
joined MSU, I was given some few other new topics for which I had to make
mhm,
PPTs. And
mhm,
you
mhm,
guys might be knowing that that if you are making a PPT, you will be having Shivratri, that you will be, pura raat jak ke you'll have to do it. So, and I mentioned this to my students also, that see, I am a human being, I am prone to make errors. So my PPTs are not Gita, Quran, Bible.
Yeah.
So I can, rather I will do mistakes, right?
Yeah.
And if I see that, obviously then it might get percolated, no, in the
Yeah.
readers who are going through those PowerPoints. So it's
Yeah.
better to always refer to the standard reference books.
I see.
Because see, ultimately, I'll be very honest, whenever I'm making PPTs,
Mm.
50% of it is copy pasted material from the reference books. Rather, I would
Yeah.
say a shortened version of it, right?
Yeah,
Mm.
yeah,
Mm.
exactly.
So it is very,
Yes, you are audible.
Okay, okay, okay. Yeah, Dhairya, I just got a message. She'll be joining back. She has some technical issue. 1%.
Okay, okay. Uh, okay, no worries.
Yeah.
So maybe I would take this opportunity, to do some marketing. So we at the SciComm Synapse, we are a group of students. I would say few PhD students from abroad and India, and few bachelors and masters students as well, working towards science communication. And it's in last like 4 years, 3 to 4 years. We had more than 22 events with different speakers, and now we would be going ahead with this kind of session where we actually invite the speaker and we discuss not just science, but also we discuss that how they reached to the point, and maybe we'll more elaborate upon the journey of the science rather than just a monotonous event. And lately we realized that our audience is also interested in this kind of interactive talk.
Yes.
So yeah,
It's
so
more of sorry, Zaria. It's more of learning from the experiences
yeah,
people have been, people
yeah,
who are in the field. So we gather those experiences and we have a good
yeah,
learning for our
yeah,
own planning, our own research journey ahead. So that's a
yeah,
good, I think, thing. Yes. Hello,
yeah.
ma'am. You are there back.
yeah, I'm sorry for the
So
interruption. My laptop got
that's
It's
discharged.
it's it's
completely
it's,
fine.
it's
Yeah,
completely fine.
we
Yeah.
were we were just marketing a bit ourselves with our audience so that
yeah,
when
okay,
we
okay.
upcome so this is something that we have started new. Earlier we used to do some events, but they were like very monotonous, I would say,
Hmm. Hmm.
and not much interactive.
Yeah,
But
correct.
now we have started, and you are our first guest.
First
Oh
guess.
my goodness. Thank you so much for this honor.
and
Uh,
and yeah, this was getting delayed since last 2 to 3 weeks due to some of the
Correct,
other
correct.
issues. So I
Right.
just told Ajinkya that doesn't matter, just let's do it and see what
Yeah.
happens.
Let's get just started with
So
it.
let's
yeah.
get started first. Yeah, so we
Correct,
are
correct.
gonna upcome with more such speakers,
Right.
and more such people. So, and we are also looking for people who can join our team,
Yeah.
because we also need resource people, we also need connection, so that we can also
Perfect.
call the professors from
Yeah.
IISc,
True.
NCBS, and of course
Right.
me and my other teammates, Surajit and other people are already here. They can also
Okay.
invite speakers from Germany and USA as
Yeah.
well.
Of course, of course.
So yes, so we are
In
gonna
field of
continue.
science is everything about connections.
Yeah,
So
exactly.
the more you people you know, more you are going to
Yeah,
grow as a person, as a
yeah,
scientist. So
yeah,
yes.
yeah, yeah. So okay, let's. So that's what I was telling to our audience, that you can stay connected with us.
Of course.
We will be
Perfect.
upcoming with more such events. So
Yeah.
this is
Sure,
just
sure.
a start of something new that I would say I have not seen anyone
Yeah.
doing.
Yeah, this is a very
So,
interesting, format that you have guys curated. So yeah, really kudos to you all.
More realistic, ma'am. I feel it's
Of
more
more
course.
realistic.
realistic. Yeah.
And I think
Yes.
I'm being realistic to the core, telling you
Yes.
everything that, yeah, I was afraid
Definitely.
of dissecting a mouse.
To be honest, here in Germany, we have to give the FELESA test. So until unless we clear this test, we are not eligible to handle mice. So
Oh.
FELESA test is something that is internationally recognized exam or a license certificate
Hmm.
which a scientist gets at any stage of life, at least after their masters,
Hmm.
maybe MBBS or Master of Sciences,
Hmm.
and then they are eligible to give the exam after 1 to 2 weeks of theoretical as well as practical training, including
Oh.
classes. And then
Yeah.
you apply for this license, you give their theory test, you
Hmm.
also give the practical test, and then you get a license that
Oh.
okay, you are recognized to handle a mice and rat.
Yeah.
Now, if you want this for frog, you have to give another fellas.
Another.
You have
Oh.
to take another license. If you want to handle monkeys, you want to handle chimpanzees, you
Hmm.
want to handle hamster, you need different licenses. So I would say this is something that we can also introduce at
Actually.
university scale,
Yeah.
at the level of university scale. So
So.
because there are many masters and bachelor students who pursue their internships, I would say, and
Hmm.
thesis, and they are sometimes afraid. So
Yeah.
if we can introduce this kind of FELISA certificate course
Right.
to young minds, then I would say it would be really good, because I was the batch where we had no experience with mice. I have never touched the mice, but now I have to do, and it's
Hmm.
so difficult for me.
Yeah, same here. For, usually I think in the previous curriculum type, they
Mhm.
had the dissection of mouse and everything in the graduation itself. But
Yeah,
again,
yeah,
mine
we
was
also
also
had.
the batch that it was ethically not allowed. So I guess
Yeah.
that's the reason why I was, not comfortable back then. Okay, today I might be able to do it, but I would
Mhm.
prefer not doing it. Somebody
Not
else
to do
to do
it.
it for me, I would be more than happy.
Yeah, yeah. Even
Yeah,
I would
really,
be
that is
happy.
really good initiative, what people have in Germany. So I think, it should be implemented in the India also.
Maybe, yeah, maybe we can start with, discussing with
Yeah,
universities and
true.
requesting universities that
Correct,
maybe you
correct.
can have some training program for
Training programs,
students
True.
exactly. Or
especially.
And yeah, go ahead, Ajinkya.
yeah, even workshops, something like mice handling or rat handling workshops, which would be really useful for people
Right,
who want
right.
to pursue their research potential in field of cancer, or any research aspect where there is animal handling is needed. Such type of workshops or trainings should would be more
Yeah.
valuable to that person.
Of course,
Yes.
of course.
These
We
So
have
that.
a message from Somilya. Uh,
Yeah,
it is like,
Yeah.
yes, I did handle mouse in my masters, but I met the mouse in very miserable condition. Of course. So, that is another
so
aspect of
I
it,
would
that it
like
is ultimate. Yeah, go ahead, go ahead.
I would like to share this experience to all, that in my master's level, I had to dissect mouse and take the immune cells out. So the mouse which I got, it was very baby mouse, around 6 days. So we had to just cut the peritoneum and tear it so that we can take the immune cells fresh. But what happened? I did the force very much, so the mouse, it tore into exactly two pieces from the
Oh my
spinal cord.
goodness. Yeah, that's what, think that's what Dhairya was mentioning about, that it
Yeah.
needs a proper training to handle
Training.
any
Yes.
system. It's similar
Yes.
to, getting an MD or MS degree, that
Yeah.
nobody
Yeah.
can just cut
Exactly.
upon a human being. So we
Yeah.
should, give that due respect to other living systems as well.
Yes.
Yes.
Yes. So that's why only we have something like a FELASA license,
Mm.
and like if you mishandle mice, and if the authorities comes to know, they can take your FELASA away. You
Yeah,
cannot anymore
fair enough.
work in void, like except like if you go out of India, China, and maybe some Asian countries,
Hmm. Hmm.
then the Falasa is not important. I would say it is required at some places, but not important. Okay.
Important. Yeah, yeah,
Yeah, it is not important. But if you step out, you need
yeah.
Falasa. Now in US, if you want to cut mice, you need a Falasa.
True, true. That is ethically also, I think, is very encouraging that,
Very.
somebody should be properly scientifically trained into it. And second thing, what I can feel is for the younger generation that obviously it is going to generate placements also. They will get job
Actually,
opportunities. That if you have cleared this exam, you have that
yeah,
specific place for you.
Yeah, you
Why
have
yes,
that
not?
skill set.
Yeah.
yeah. Maybe a technician's job funded by
Exactly,
DBT.
exactly. Because I'll tell you, in IIT Bombay, obviously not for the animal system or all, but
Mhm,
for let's say fax analysis, for
mhm,
electron microscopes, we do have
mhm.
assigned technicians to it.
Yeah,
So even if
yeah,
I
yeah,
want to analyze my sample, I'm not allowed to touch that machine. I can be in the room,
yeah.
I can interact with
Mm,
the technician, but the
mm, mm,
technician is the only person who is going to handle the instrument. And
mm,
it has
mm,
two basically important points. What I feel, first thing is that the instrument is maintained in a well condition. Otherwise
mm.
anybody can
I
come,
see.
they have either half knowledge or no knowledge, they will try to tinker around with the instrument, the instrument
Mm,
is going to go bad. And
mm,
second thing
mm,
is obviously we are generating the job opportunities. So
mm.
both ways I think it's a win-win situation.
Hmm, I see. So I also had one more. Maybe next time I can get him for the speaker. He's my very good friend. Maybe your batch only. He was working at IIT Bombay. He was a MSU graduate from biochemistry. He was
Okay.
working in the field of NMR,
Acha.
and then later on he dropped IIT Bombay. He was working there for 3 years. It continued,
Oh.
and then he dropped the lab, and then because he got into a lab who is like godfather of NMR,
Hmm.
so in Germany. So maybe we'll also have him and his experience about
Yeah,
IIT, and
correct,
how's
correct.
the culture when you start from a very middle class family and
Hmm.
your destination, how you reach there, right?
Correct.
So now so now people are also waiting and eager to listen about
Yeah.
your research and
Okay, okay.
what you exactly did in your PhD. So I would like to take this opportunity and welcome you again
Thank you so
to
much.
give a short presentation on
Yeah.
your work on during your PhD, and maybe you can keep it as simple as
Yeah,
possible
yeah, sure, sure.
as
I will try to
our
be,
view.
talking in very
Yeah,
basic language which everybody would be able
yes,
to connect with. I'll try that.
yeah, yeah, exactly. So we also have a few of our, just a second, we also have few of our participants at YouTube live. So maybe for them, if you have any questions,
Yeah.
you can put it in the chat box. And also the audience that is here, you can also write your questions in the chat box. Later on, me or Ajinkya would take those up,
Yeah.
and we would take it up to Dr. Jaswandi later. Okay, ma'am, all yours.
Sure, sure. Okay.
Yes.
So yeah, before beginning my presentation, today is
Yeah.
17th September is usually celebrated as International Microorganism Day. So
Ah,
yeah, let's dedicate today's talk to microorganisms, highly
that's why
intelligent.
you chose 17th September
Yeah.
when you were offered the date.
No, that
That's
Ajinkya
really
offered me, so thanks to him.
okay. Ajinkya, that's a
Okay.
sweet of you. Okay,
Chalo, shall I start?
let's start. Yeah.
Yes,
Okay,
yes.
okay.
Let's start.
Okay, I hope my screen is visible to all of you.
Yes, ma'am. It's
Yeah,
visible. Could
great.
you just make it in the PowerPoint mode?
Yeah, I'll go do that.
Yes. Technical
Okay.
team can put it on the pin, and so that our YouTube viewers can also join. And next time, maybe whenever we do our events, even next time, please don't hesitate to also join on YouTube, because getting into the meeting, Google meeting, is really sometimes disturbing to speaker as well. We have seen that people come and go, and that was the reason we chose YouTube as a platform for all the events. So exactly, it's the same. It's nothing different on YouTube as well. So yeah, you can also next time also, if you get a YouTube live link, please do not hesitate to join. Okay, thank you. All yours.
Yeah,
Sorry.
okay, great. So I will not take much of a time. I will try to be as crisp as possible and as simple as possible. And still, if somebody has some questions, maybe you can write it in the chat box regarding the talk also, and I would like to answer them afterwards. Is that okay? If that works for everybody?
Yes.
Okay,
Yes,
so
yes,
I'll be
yes.
talking obviously about the yeast system, that is a surrogate system to study tumorigenesis. Now, just a brief introduction that obviously yeast system has been used as a model system for various reasons. That, and one of the important one is it has almost 23% of the genes homologous to the humans. So, for example, the apoptotic pathway, the biochemical pathways, metabolic pathways, most of the pathways and genes involved into it, they are very much conserved in the yeast and human system. So that makes a very good model system for the yeast. Obviously, it is a very good expression system. Being eukaryotic system, it will have all the post-translational modifications and everything what might be necessary for the proteins. And obviously, it has a very compact genome, 16 chromosomes, and thankfully all of the genome sequence is available, curated, available publicly to everybody, and it is a very easy system to manipulate. As I mentioned, though it is an eukaryotic system, handling yeast cells is very much similar to that of the bacterial system. Their generation time is also 24 hours to 72 hours depending upon which carbon source you are using for their growth. So that makes yeast a very good model system, and hence it has been used to study various aspects of human diseases and metabolism. So many of you might be knowing that is the CDC proteins, the cell division cycle proteins, which are involved into cell cycle regulation. Most of them were first identified into the yeast system, Saccharomyces, and then people carried out further studies and found that they have the homologs in the human system as well. So obviously they have been used to study the mitochondrial disorders. Sorry for this mistake, it has to be only one, the mitochondrial disorders, neurodegenerative disorders. For example, Parkinson's. Nowadays people are more focused on using yeast as a system to study diseases like Parkinson's disease, Alzheimer disease, which is because of the accumulation or aggregation of the proteins. And a group from IIT Bombay itself, Samari Maji's group, they have worked with this. They have used P53, again P53 protein, and they have studied its aggregation, how it affects the yeast system and all, which can be further reciprocated to the human system. And obviously, the yeast has been used since ages to study the cancer. And today I will be focusing onto that aspect of studies. So, in 1926, a scientist called Warburg, he had proposed that in normal cells, as you can see, a glucose is converted to pyruvate via glycolytic pathway. Then it is further fed into mitochondrial system through TCA cycle to generate the energy. But what Warburg found out was in tumor cells, the glucose is converted to pyruvate, but then it is converted to lactate. Most of the cancer cells, tumor cells, they have this characteristic property, that is of fermentative metabolism, where glucose is converted to lactate and not completely oxidized to the carbon dioxide and water. And obviously this phenomenon was known as the Warburg effect, correct? And this Warburg effect, people then started that why might this be happening. So there can be two aspects for it. The first one is the mitochondria is defective. So obviously whatever pyruvate is synthesized cannot enter the TCA cycle, and hence the enzymes convert that to lactate in basically the tumor cells. And other thing can be because of the hypoxic condition, that is low oxygen conditions, right? But in last okay, so to give explanation to Warburg's effect, a scientist called Crabtree, what he showed was, again in tumor cell, that was in 1928 if I am not wrong, that tumor cells, they take up lot more glucose, they internalize lot more glucose as compared to the normal cell. And this excessive amount of intracellular glucose, it basically inhibits the mitochondria. So mitochondria are not defective, it's just that their function is inhibited because of some or the other factor. And this particular phenomenon obviously is known as the Crabtree effect, right? And in last decade, to support Crabtree effect, people have found that in tumor cells, it is not necessary that the mitochondria are defective. They have isolated the functional mitochondria. They have done studies in right models, and they have also shown that the oxygen conditions are also not hypoxic, they are normoxic conditions, but still cancer cells are showing this particular property of the aerobic fermentation. So why everything, this is happening? So basically, what is the connection between this Warburg effect and the Crabtree effect? Okay, so this is basically the I would say majority of the main properties of the tumor cells, that they show aerobic glycolysis, that is the fermentative mode. And another important property is they evade the apoptosis, that means they become immortal. They escape apoptotic pathway, but the exact relationship between them was not known back then. That means whether the ferment cell become metabolically inclined that way, that it will have only fermentative mode, and if that leads to the evasion of apoptosis, or there is some defect in the apoptotic pathway because of which the cell becomes adopts to the fermentative pathway. It's like a chicken and hen story. So we wanted to find out if there is any connection in between these two or what it is. So for that, we chose a p53 molecule, which is the key regulatory molecule. And the reason behind choosing the p53 is, as we all know, it responds to various signals from the cell. It might be DNA damage, oncogene activation, low oxygen conditions, etc. And then p53 being the transcriptional activator, it initiates the transcription of various genes, the products of which are involved into these many aspects, that is DNA repair, cell cycle arrest, anti-angiogenesis, apoptosis. And in last decade, it has shown that p53 has a major role to play in regulation of carbon metabolism as well. That means in normal cells, the p53 will promote oxidative phosphorylation, and obviously if there is any mutation in p53, that regulation would be revoked. And that's what exactly happens in the tumor cells, right? In tumor cells, p53 is the most abundantly mutated protein. Majority of the cancer cells, they do have mutation in p53 along with the other mutations. So again, we have this common link with p53, that p53 has a role to play in both the regulation of carbon metabolism as well as the apoptosis. That means the fermentative metabolism as well as evasion of apoptosis. So that's why we chose p53 as a candidate molecule to be expressed into the yeast system. Now, yeast system, specifically the Saccharomyces cerevisiae, it has the fermentative mode of carbon metabolism even in the presence of oxygen. That's why Saccharomyces are very commonly used for wine production, ethanol production. So they ferment glucose into the alcohol even if oxygen is present. The second important point for choosing yeast as a model was the apoptotic pathway of yeast and humans is very well studied, and it has been shown that it is very well conserved as well. And the another important point was the homologue of p53 is absent in yeast. So when we express human p53 into the yeast system, it basically works as a clean slate to study whatever effect the p53 might have onto the cells. So that was the reason for choosing p53, first of all as a key regulatory molecule for cancer as such, and second using the yeast system. So now what happens into the yeast system? This we have seen in the human cells, that the tumor cells basically, they convert glucose to lactate, and it does not go through the mitochondrial phosphated oxidation. And glucose, excessive internal glucose, it inhibits the mitochondria, which is known as the Crabtree effect. In yeast also, the Warburg effect, what we can say is exists, but instead of lactate, the glucose is converted to ethanol, that I mentioned already. And Crabtree effect is very well known as glucose repression in yeast. So we can see there is a parallel between yeast system and a human system, and it forms the very good model system to study the human diseases. Now, to further elaborate why we are using yeast and what kind of yeast we have chosen. So in my study, I have used two genera of yeast species. That is, one is Saccharomyces cerevisiae, and one is the Clueromyces lactis. So the major difference between them is the Saccharomyces cerevisiae is a Crabtree positive yeast. That means excessive intracellular glucose is going to inhibit the mitochondrial function, whereas the K lactis is a Crabtree negative yeast, where even if there is excessive intracellular glucose, the mitochondrial function will go on. So I would like you to remember this point because that will be coming up in the next slides also. So Saccharomyces cerevisiae is a Crabtree positive yeast, whereas K. lactis is a Crabtree negative yeast. Another important point is the Saccharomyces ferment the glucose to ethanol, which accumulates in the medium. That's why, again, as I mentioned, it is used in the wineries. Whereas in K. lactis, it though ferment the glucose to ethanol, but it does not get accumulated in the system. As in when ethanol is formed, it is further metabolized through other pathways, and there is no accumulation of the ethanol as such. And the third important point is in Saccharomyces, we can have the peptide strain. That means the mitochondria are dispensable. That means even if mitochondria are removed from the cell, the Saccharomyces is able to survive on sugars like, let's say, glucose. However, in K. lactis, the mitochondria are essential for the survival. So here itself, I will tell you that we use Saccharomyces cerevisiae as a model for tumor cells, whereas K. lactis as a model for the normal cell. I know this is the very vague parallels that we have drawn, but it fits perfectly with the phenotype what tumor cell and normal cell shows. So S. cerevisiae basically represents tumor cell and K. lactis represents the normal cells. Now, summarizing the introduction that I have talked so far to gain insight into the role of fermentative metabolism, Crabtree effect, and its relation with mitochondrial role, if any, that exists in p53 mediated apoptosis. We studied the effect of expression of p53 in S. cerevisiae, which is a Crabtree positive yeast, and K. lactis, which is a Crabtree negative yeast. Okay, so we started our work with the yeast system. I'll just give you a brief idea that in yeast system, there is a pathway for galactose metabolism, and we have chosen an yeast system which is first capable of metabolizing galactose. The pathway is entirely intact, and in that system, we have expressed the human p53 protein from the galactose inducible promoter. Correct. So this is the one type of yeast that we have used. And in another one, what we have done is we have deleted the GAL1 gene, which is the galactokinase, the first enzyme of the galactose metabolism pathway. And in that strain also, we have tried to express the p53 from the, again, galactose inducible promoter. So these are the two systems that we have used. And just to give a summary of whatever strains we have used in the study is the GAL1 deleted strain, which cannot metabolize galactose, but it can grow on the other sources and it shows Crabtree effect. The GAL1 positive cell, these two basically represent the Saccharomyces cerevisiae, whereas this one is the KL-GAL1 represent the K. lactis strain. So both of them can grow on galactose, can grow on other sources also, but the major difference is K. lactis is the Crabtree negative strain, right? So this is a diagram where what we have done is we have grown the cells into a liquid medium, and then we have plated onto a medium containing glycerol as a carbon source. So whatever spots you can see is basically the tenfold dilution of the culture. So we start with 0.5 OD, and then we serially dilute that, and a particular amount, let's say 5 microliters, is spotted onto the plate. So this is how, you know, this particular represent one strain, this is another strain, so on and so forth, right? So this was the strain. This is GAL1 deletion strain. You can see it is growing onto the glycerol. This is GAL1 deletion strain where p53 is expressed, and obviously the medium has galactose because the p53 is expressed from the galactose inducible promoter. So moment you add galactose into it, the genes are going to get expressed, whatever are downstream to the promoter. So in GAL1 deletion strain, which cannot metabolize galactose, it has to rely on to the glycerol, which is a non-fermentable carbon source, where the involvement of mitochondria is necessary for this metabolism. There we can see the expression of p53, which is very much evident here, is showing the growth inhibition phenotype. Whereas in the same genetic background, if the GAL1 gene is intact, that means the cell can metabolize galactose. So even if it is the glycerol and galactose, the cell is going to prefer the galactose because that is a fermentable carbon source. Any cell prefers fermentable carbon source for metabolism. And in that case, even though p53 is expressed, there is no inhibition of the growth that is seen. So this was very interesting observation that we found. And then we further try to look for the same strategy onto the other carbon source. So now instead of glycerol, we have used sucrose as a carbon source. Now again, the major difference between them is the glycerol is non-fermentable carbon source, whereas sucrose is the fermentable carbon source. But here, interestingly, the same strain, the Galvan deletion strain with P53 expression into it, which was unable to grow on the non-fermentable carbon source, is now happily growing onto the fermentable carbon source, even though P53 is expressed. So from this, we conclude that the growth inhibition due to the P53 expression depends on to the mode of metabolism followed by the cell. So if it is non-fermentable carbon source, mitochondrial involvement is necessary. It is showing the effect, and the cell are basically not able to grow. It is inhibiting the growth. Whereas if it is fermentable carbon source, no need of the involvement of the mitochondria, and hence the cell can happily grow. The similar studies we have carried out in the liquid medium also. I'll just quickly go through this. So that was the plate assay which we have done into the liquid, and there also we saw similar results. That is, the Galvan deletion strain expressing P53 was able to grow when there was no induction of P53, but when there was induction of P53, we can see the CFU number is low. On the other hand, the strain which is expressing P53 but can also metabolize galactose has hardly any effect onto its growth, right? Even though P53 is expressed. And now talking about K-lactis, now this was very interesting to us. So again, we took K-lactis strain. There also we have expressed P53 from the Galvan promoter, the galactose inducible promoter. This I have already talked about. This is the Saccharomyces strain, where the wild type strain which can metabolize galactose does not show any growth inhibition, even though the P53 is expressed. Whereas, interestingly, the K-lactis strain, which is, remember, it is a Crabtree negative strain, where even though it can metabolize galactose, okay, so this strain background is similar. We can say metabolically, this strain, the Saccharomyces strain and K-lactis strain, metabolically they are similar, but still the expression of P53 is having the growth inhibition effect onto the K-lactis strain. And that is only because of their preference for utilization of the mitochondria during metabolism. Correct? Now, so obviously we have done the studies on the other carbon sources also. I will not go into the details of that. That is similar. That is K-lactis. The growth inhibition is independent of the carbon source that has been used. Whereas in Saccharomyces cerevisiae, it depends on which carbon source is used, because then ultimately the cell decides whether mitochondria has to be involved into the metabolism or not. Right? Now, somebody can comment that, okay, this is, human P53 that you're expressing into the yeast, that is a foreign protein for the cell. It might have, the burden onto the cell, and that's why the cell are getting killed. So to rule out that possibility, what we did was we chose different mutants of P53, which are very well established to be mutated in the cancer cells as well. So we have done site-directed mutagenesis in the wild type P53 gene, and we generated these mutants. And there also we can show that few of the mutants, for example, this is again the Galvan deletion strain, and we can see it is showing the growth inhibition phenotype. I think, yeah, I have the Western blot for it also. So we can see these mutants also, they are showing the same phenotype, which is shown by the wild type P53. So that means the effect, the growth inhibition effect that we are seeing is not because just any random protein getting expressed, but the functional P53 has to be present in order to show its inhibition effect. So again, that was a very interesting phenotype. And similar studies we have done in the K-lactis also. There also we showed that functional P53 is essential. Okay. Now, I started my studies in 2010, and in 2011 there was a group who showed that expression of P53, they actually induce the apoptosis into the cell. They have shown the different markers of apoptosis when P53 is expressed into the cell. So we wanted to see if that is also the case in our system also. So for that, we have studied the reactive oxygen species level. We have also looked for the exposure of phosphatidylserine, which is the characteristic of cells undergoing apoptosis or the programmed cell death. And we also look for the increased oxygen uptake. So for the strategy for that is, we again grew the cells into the glycerol, because that is the non-fermentable and the neutral carbon source. This was from the colony. Then we made the inoculum out of it, and that was again inoculated into the fresh medium, where the one system was only glycerol containing medium that will serve as a negative control. Then in another system, we have glycerol and we use H2O2. Basically, that is the inducer for the apoptosis, the hydrogen peroxide that will act as the positive control. And then in another system, we incorporated galactose that is going to induce the expression of P53. And now these strains, other cultures, they were further analyzed for subjected to annexin staining to look for the apoptotic markers. It was used for ROS determination and it was used for the oxygen consumption. So obviously, as we can see, this is basically the representative image for the annexin and PI staining. So the cells which are in the initial stage of apoptosis, their phosphatidylserine is exposed outside the membrane and it is stained with the annexin dye. The PI, basically propydium iodide, it stains any dead cell. It can be apoptotically dead or the necrolytic cells also. So we should look for the green signals and not the red signals. And I have actually counted the cells, 1000 cells per strain, and we have got this data. So as we can see now, this strain again, the Galvan deletion cell of Saccharomyces cerevisiae, which was showing growth inhibition effect upon P53 expression, has higher number of dead cells which are showing the apoptotic marker, and similar with the K-lactis strain, it is showing increase in the dead number of cells upon expression of P53. And this was for the cells which are grown in glycerol, and this was for the cells which were grown in sucrose. So by this we confirm that yes, in our system also the P53 is causing apoptosis, but if you look from the strain background point of view, it is only to the strains which are showing growth inhibition phenotype and not the other cells. For example, this, the Saccharomyces cerevisiae wild type strain for galactose utilization. It did not show any growth inhibition phenotype, and here also it is not having any cells which are undergoing apoptosis because of the P53. Next, we determine the reactive oxygen species level. That is the spectrophlorometric analysis, and there also the similar result was found. That is, the strains which are undergoing growth inhibition phenotype and were showing apoptotic marker, they had higher levels of reactive oxygen species. That is the Saccharomyces cerevisiae Galvan deletion strain and K-lactis wild type strain when grown in glycerol, and similarly when grown into the sucrose. And third, what we looked for is the rate of oxygen consumption. This is using the Clark's type of electrode. There also similar results, that is the strains which are showing growth inhibition phenotype had higher rate of oxygen consumption as compared to the other strains. Right. So all these studies together led us to the conclusion that in our cell, okay, so to further add on to it, as I mentioned earlier, that in yeast and humans, the apoptotic pathways are very much similar and conserved. So I chose few yeast genes which are involved into apoptosis, like MCA1, AIF1, NMA111, and NUC1, and their respective orthologs of the humans I have already mentioned here. And what I did was I deleted these genes individually in the strain background, which was expressing the P53. So basically, we wanted to know that P53, when it is expressed into the yeast cell, does it have any interaction with the apoptotic genes of the yeast as such or no? Because in humans, we know that P53 has a role to play in activation of these apoptotic genes. So we wanted to know if this similar case happens in the yeast as well. And interestingly, we found that after deletion of AIF1 in this yeast, which is the mitochondrial protein and involved in the DNA degradation, whose human homolog is AIF, that is apoptosis inducing factor, we could see when this gene was deleted into the Saccharomyces cerevisiae background, showing growth inhibition phenotype, the inhibition was marginally rescued. Obviously, it did not rescue the growth inhibition completely. There was some marginal rescue for the phenotype, and that can be attributed with the fact that apart from AIF, some other genes might also be involved into this. But this gave us very good insight into the point that the human P53, which does not have any ortholog into the yeast, it has the capability to interact with the proteins of the yeast origin. Might be in a similar fashion that it is doing into the human system. So that was a very good observation that we had. So To quickly summarize what I have discussed so far, the p53 expression leads to growth inhibition in Saccharomyces cerevisiae only on non-fermentable carbon source where involvement of mitochondria is necessary and not on fermentable carbon source, whereas overexpression of p53 leads to cell death in K-lactis, which is a Crabtree negativist, independent of the carbon source, because in that strain, mitochondria are constantly active, which is very much similar to that of the normal cells of the human. And hence this indicates that the fermentative mode of metabolism per se is not essential, or it does not play a role in escape from apoptosis. Rather, the mitochondrial state, the functionality of the mitochondria is essential, and that would be decided by the Crabtree effect. So Crabtree effect is very crucial, and usually fermentation per se is not essential for the apoptosis, right? So that's what we conclude. Now, to conclude my talk, I would again would like to thank the organizers, Ajinkya, and everybody else, and of course, a great gratitude to my guide, Professor Jaidev Bhatt. He is standing here. This is in front of the IIT building. What we have split our picture. These are all my lab mates, and of course, Dr. Abhay Kumar, who was my senior in IIT Bombay, who initiated this work, and then once his doctoral degree was done, I carried forward the work ahead, and of course the CSR and IIT Bombay for the financial support. Yeah, that's all from my side.
That was really intriguing for, I would say, all of us, and especially for me, because uh, my project is on not p53, but
Yeah,
when there is an inhibition of apoptosis,
correct.
uh, and when there is a virus induction in these
Hmm.
cells,
Hmm.
then ideally the pathway should not go through p53,
Yeah,
but
true.
ideally, but in my case I am also seeing some down regulation of p53, which is making me confused that why is this happening. I mean, p53 is not at all in the picture of my pathway. That there are
Correct.
many cancer pathways and
Right.
many cell death pathways, so my pathway has nothing to do with p53, but still I'm getting some p53 vibes,
Right.
uh, and the main protein that I'm working on, that is Puma,
Ah.
is quite tricky, I would say.
Yeah, of course, of course. That's what, no, P53, no doubt people have worked so much with
Yeah,
P53, but
yeah,
still you never know what, P53 has in store to serve more.
yeah. I would say it is a multitasking women
Correct. Yes.
doing everything
Yeah.
here,
That's
there,
what, no, it is
and
involved into cell cycle regulation, apoptosis,
yeah,
carbon metabolism, whatnot.
yeah, yeah. So it's like a multitasking mother, I would say, of the cell, doing everything,
Yeah.
wants to do everything.
Right, right.
So, so maybe I would request participants to first add their questions in their chat box,
Chat box,
in the meeting, as well as in the uh, Google meeting, as well as in the YouTube live.
YouTube live.
So yeah, so you can please write your questions. It can be generic questions, it could be specific questions.
Big
So
questions.
please, you can have us.
Yeah, anybody. I hope people have understood what I have worked on.
Yeah, maybe if you
Yes.
can, since I am from a bit yeast background, I
Yeah,
worked on yeast, but I never worked on growth assays and
correct.
things like, not growth assays, death assays, or what do you
Yeah.
say?
Apoptosis,
Death assays.
growth inhibition. Yeah,
Growth.
Growth inhibition. So
correct.
maybe can you quickly, summarize method that how you perform this assay? Because I know we take multi-channel pipette and do many things, but I think the audience here, they maybe some of them have worked with the death assays, or I would say,
Right, right.
but like, what do you exactly
Okay,
do in that as a
right,
methodology?
right.
So it
So,
would be good.
obviously, whenever you guys might be knowing, whenever we work into a laboratory, the one thing leads to the other. So, obviously, the first point was we saw the phenotype that, okay, because of P53 expression, the cells are not able to grow in some particular condition. For that, it was a glycerol containing medium. So then we wanted to know, as you know, the classical microbiological experiment goes, let's look at this colony forming units. So we did the growth curve analysis and tried to look for the OD and corresponding colony forming units. So there also we could see that because of P53 expression, the CFU number was low as compared to the other strain where there was no P53 expression. That was second point. Thirdly, I have not shown that data, but what I did was I simply did the propydium iodide staining just to see if cells are dying or it is just the static inhibition of the growth. That means if cell can be reverted back if that P53 system or the environment is removed. But of course, in the propydium iodide analysis, I could see that cells are actually getting killed. The It was positive for the PI staining. So that gave us the insight. Okay, it is not just the static inhibition of the growth, it is actually the death of the cell. So initially I have used the word inhibition because at that time it was not very clear whether it is death or just the inhibition. But eventually we got to know that okay, it is actually the death of the cells. And then death also can have either it can be necrotic death or the programmed cell death. So again, to distinguish between them, I went ahead with the antigen staining, which is very commonly used to study apoptosis. So that's how
Yeah.
it was the flow of the experiments that we
So
designed.
it was apoptosis.
Yeah, correct. Yes.
So it's a programmed cell death.
Right.
So
Right.
I work on, especially I'm interested because I work on the switch, that is switch
Yes.
between necroptosis and apoptosis, and
Right.
intervention of RIPK3, caspase 8,
Right.
and especially Puma and
True. True.
BCL2 family proteins. Yeah, yeah.
True.
So that's major of something that many of them, the field is also changing in the
Of
cancer
course.
research as well, that like people are now also considering necroptosis as a program cell
Yes.
death.
Yes.
People
Yes.
are considering, I mean,
Of course.
I have read recent papers after, I would say, 2019 in the
Hmm.
cancer research.
Hmm.
People have mentioned, they have not exactly said that it's a necroptosis or apoptosis. They have told that it's a program cell death, which
Yes.
includes necroptosis as well,
As
and
well.
which
True.
was
True.
something, which was something very interesting for me, because when we studied back in like 2019-20, then it was
Correct.
differentiated that okay, program cell death is apoptosis and autophagy, and
Hmm.
now if you are talking about necroptosis, then it
Hmm.
is something which is not program cell death.
It's not
But
the
now,
conventional method, what we
yeah,
can
the
call it as.
conventional rule,
Yeah.
but now the cancer research and the cell death research is completely shifting towards this structure where
Different. Yes. Yes. Yes.
yeah, and there I see the advancement of science, that oh, I studied this, and now I am working on this, which is like advanced version of what I
Yeah.
studied. So it gives me really, this is the beauty of the research, I would
Science.
say,
Of course. Of
that
course. Yes. Yes. True.
everyone
That's
wants
what
to
it
do.
it also depends on somebody's
Yeah.
perspective. That obviously in
Mm,
our research also, whatever we have thought, that somebody might have different, altogether different view on it.
mm,
They might come up
mm,
with some different experiments to prove the same
mm,
thing. So that's
mm.
really very fantastic of science.
I see. Maybe let's go with some basic questions
I
first.
think, yeah, we have few
There are,
questions.
yeah,
We
yeah.
have lot many questions.
One of them is asking on YouTube that why, like, how will you explain the P53 involvement in cancer, like at the very basic molecular mechanism of P53, like
Yeah.
what is what P53 does exactly
Okay.
in the cancer?
True. So, if you remember the initial slides, I have mentioned that in normal cells, we'll just first talk about the normal cells in the normal human being. The P53 is a transcriptional activator basically, and it is regulating the expression of genes involved into various pathways. It is metabolic pathway, it is regulation of the cell cycle, that means how frequently the cell should divide, and it is also regulating the DNA repair mechanisms and etc. Right? So its function is very much necessary to maintain the homeostasis in the cell. Now, in cancer cell, what happens, as we know, there is some mutation in the P53, and because of that mutation, this P53 is now deviates from its normal function. That means it will not be able to regulate these systems and maintain the homeostasis or harmony. And that might lead to, let's say, if it cannot maintain the cell cycle, then obviously the cell are going to divide rapidly, and that is going to form a tumor cell as such. If it cannot control the apoptosis, even if there is some damage into the DNA, that is going to get transferred to the other cells. It is going to proliferate, and that is going to give you the mutated cells, which is again going to form a tumor. So that's how the P53 has a major, rather I would say, the one of the important roles to play in tumorigenesis.
I see, I see,
Yeah.
I see. So one of the participant is, asking that how is yeast correlated with neurodegenerative disease? I would like to say
Yeah.
something on this because I was
Okay, go ahead.
working on
Yeah.
something similar, and then I'll of course tell ma'am to go ahead. So I may be wrong. What was my experience is that we take yeast and we give oxidative stress by various means. Like I used to use sodium azide, and few also they used H2O2 or other oxidative stress inducing agents like etoposide as well, right?
Correct, correct.
So in the neurodegenerative disease, it is mostly that your RNA and your proteins are trapped into P bodies and stress granules, which is like very recent finding after 2012. I would say if you are connected in the
Mm.
field, then
Mm.
majority of the neurodegenerative diseases show these P bodies and stress granules in the cells, in neuronal cells as well. And people use this yeast system as a model to study P bodies and stress granules. And where I was working at IISc in this, so I was working on like formation of P bodies and stress granules because my guide had a pioneering lab which worked on this. So that those P bodies and stress granules are then correlated with the human neuronal cells
Right.
later on.
Right.
Maybe then they work with neuronal cultures as well. But
Correct.
initially, if you want to see these stress granules, they started with yeast
True.
to monitor this. So maybe,
Right.
ma'am, you can
No, Dharya
shed
explained
more
it
light.
perfectly. I would just add to that, the practical aspect of it. Obviously, as you said, the
Mhm,
P bodies and everything. So
mhm,
it is very easy to manipulate the yeast system so that even if
mhm,
something of this sort is happening, it is easy to decipher. No doubt, you
mhm,
can
mhm,
work with the mammalian system as well, but you might be knowing the integrities that comes with handling the mammalian system. You have to have specialized laboratories. You have to have
mhm,
that
mhm,
medium, serum, everything.
mhm,
But yeast cell,
mhm.
even simple glucose and nitrogen, they're good to go.
Then
So
also
that,
they don't grow sometimes.
yeah, exactly. So yeah, those are things to be ignored sometimes. That is part
Yeah,
and parcel of research life.
yes, sometimes they grow, and in the morning you go, the culture is not ready.
Yeah,
So
exactly. See,
it
we are,
happens.
I always tell to my students that we are at microorganisms' mercy. If they want to give us results, they will give. Otherwise, you will do everything, nothing is going to happen.
So we have one more question which
Yeah,
says, can you please shed some more light on reactive oxygen species
yeah, okay.
and its connection with the Crabtree effect, and how exactly they are correlated with each other?
Okay, so see, basically, I don't see, rather, I don't know if there is any direct correlation between them. But obviously,
Mm-hmm.
see, Crabtree effect is there is excessive of glucose that is inside the cell,
Mm-hmm.
and that is somehow is going to inhibit the mitochondrial function. Correct? And if, as we all know, if there is some impairment in the mitochondrial function, there is going to be increase in the reactive oxygen species, superoxides, everything. So, to be very frank, I do not know the exact correlation between them, but yes, they have some, things to do with each other. So, yeah, again, I think it boils down to the intracellular glucose concentration, if we have to talk from that perspective.
I see. I
Yeah.
see. So I think
Yeah, I think we have just a second. We have one more question, that how does cancer happen at molecular level? So that is, I think,
okay.
question from YouTube. Okay, that is a very difficult question to answer because
A lot of things.
yeah. So that's what, every cancer is unique, and I would say every cancer patient is also very unique, because whatever mutations or whatever metabolic changes a particular cancer patient has, it need not be necessary that another fellow having similar cancer, let's consider breast cancer for example, two individuals might have totally different mutations, totally different mode of metabolism, in the sense which thing is preferred, which is not preferred, which enzyme is more reactive, which is not more reactive. So this is very difficult to come to one concluding point, and I guess that is the major drawback, and major, I would say, the halting point in treatment of the cancers. That's why people are nowadays are shifting to personalized medicines, where you will have to study an individual completely and then come up with some medicine which would be suitable to that particular person, right? So yeah, but to answer that question in very layman's language is that at molecular level, obviously a single mutation is not going to lead to the cancer. There has to be more than one mutations, so that, let's say, one mutation is in the cells which prevent them from undergoing apoptosis, another might be preventing them from utilizing the oxidative metabolic pathway, and as a whole, to sum up those mutations together, it might lead to cancer. See, always any tumor need not be also cancerous tumor, it can be the benign tumor. But if anything else happens to it, again, the lifestyle also, whatever you eat, whatever you're putting into your system, that also has a major role to play into this. So it's very complex system.
Definitely,
Yeah.
definitely. So we have more questions as well. So
Okay.
Dhara is asking that P53 has
Huh.
4 subunits,
Correct.
and as we know that it is an example of proto-oncogene.
Hmm.
So mutation in only one copy of gene. You mean one subunit? I think so. She meant one
No, but
subunit.
that is, P53 is a homopolymer. So okay,
Okay.
go ahead, go ahead.
So mutation in only one copy of gene leads to mutation. The mechanism is the same in humans and yeast both.
Yeah, so to answer her question, I can say in East, to be specific, in our system, it was only single gene copy that we had cloned. It was integrated into the genome. Correct? The whatever might be a wild type P53 or a mutated P53, it was single copy of the gene integrated into the genome. So obviously, whatever
Hmm.
protein we get will be from that single gene. So yes,
Hmm.
to answer her question, it is the same mutation, all the
Hmm.
mm, polymers.
Hmm.
Yeah.
I see. So we have one more question. Why we consider GLACT instead of normal human cell to study about P53 mechanism in a tumor cell?
I'm not very clear about this question, but I think she wants to ask that why we have drawn parallel between K lactis and normal cells. I guess that's what she wants to ask. So I will ask
Hmm.
on that aspect, that as I mentioned, that Saccharomyces can throw away the mitochondria and become petite strain, which is
Hmm.
usually we can consider as, let's say, dysfunctioning of mitochondria. Even it,
Hmm.
either absence or non-functionality can be considered similar. So Saccharomyces can be correlated to the tumor cells, where mitochondria, we say, might not be taking part into it, whether it is present or absent. Whereas in Cluromyces lactis, the another yeast species, it has to have mitochondria all the time, whether it is utilizing any of the carbon source. And that's what the normal cells are doing exactly. We cannot just our human cells just cannot throw away mitochondria and live happily. That cannot happen. So that's why we have drawn these parallels between SRVC and K lactis. I think that's what she wanted to ask.
So we have one more question.
Yeah.
We'll just take up quickly.
Yeah.
So it's the c-MYC transcriptional factor, which is responsible for cell proliferation.
Hmm.
So does P53 play a role to regulate the activity of the c-MYC for the cell proliferation process, or in both normal and cancer cells?
See, in normal cells, obviously it has a role to play, right?
Mm,
But in mutant
yeah.
cell now again, in cancer cell rather, basically, it will depend
Mm,
if that particular cancer cell has a mutation in p53 or no, right? So
mm,
obviously I
mm,
mentioned
mm,
that most of the cancer cells, they do have mutation in p53, but there are
mm,
few cancer cells which might
mm,
not have mutation in p53 and
mm,
have mutation
mm,
somewhere else, right?
mm.
So I think it's only 50 or 60% of the cancer types which has mutation in p53. Rest of them,
Mm.
p53 is absolutely normal, but something is wrong somewhere else, right? So it
Yeah.
depends, it is context based that which cell particular
Mm.
we are talking about, but of
Yeah.
course it has a role to play in semic activity.
Uh-huh. Uh-huh. I see, I see. So maybe continuing in this question, as you
Yeah,
mentioned that P53 is not the case in all the cancers. So
correct.
can you tell if there are some known cancer where P53 doesn't play, or maybe some phenotype, I would rather say, some tissue phenotype where P53 is not involved, and like how can you approach this, because whenever you see any cancer paper they have at least done a western blot for
Of
p53,
course. True,
at
true,
least.
true, true.
So I have seen many cancer papers and also cell death papers, and majority of that they do p53, irrespective to the antibody not working very
Correct.
great for p53, but
Right,
still everyone tries to get p53. So
right.
what in the case where you just delete p53? Even I had this question earlier. If you
Hmm.
just delete p53, then what?
Okay, so uh, I have not come across any paper which talks about complete deletion of p53, but if
Yeah,
you look
yeah,
from the the logical point of view, obviously if p53 is not there, the functional
the animal
protein.
will die.
Yes,
Yeah,
exactly. So there would be no survival as such, and that's
yeah,
why
yeah.
I think the mutants of p53, they are more prevalent, or I would say they are
Mm-hmm,
detectable in the
mm-hmm,
population. That okay, there is mutation in
mm-hmm,
p53, and
mm-hmm,
uh, the candidate is surviving with some disease, and hence the sample is available for analysis, right? And to answer your question, Dhairya, that which particular cancer type which does not have p53 mutation, so I
mm-hmm,
think as per my knowledge, it is not again restricted to any particular cancer type. Rather, it
mm-hmm,
is dependent on an individual itself.
mm-hmm,
That
mm-hmm,
you can take uh, let's say candidates from all the cancer types,
mm-hmm,
majority of them, irrespective of the cancer what they are suffering through, will have mutation in p53, but few of them might not have mutation in p53,
mm-hmm,
right? So
mm-hmm,
it's not that
mm-hmm,
okay, this, let's say pancreatic cancer, they will not have mutation in p53. No, it's not like that.
mm-hmm,
It's
mm-hmm,
on the individual, whether that particular
mm-hmm.
individual cell, does it have mutation in p53 or no.
Ah, okay,
So again,
okay.
the complexity is to another level.
Yeah, I mean, it's quite difficult. But this is something that I have always seen. Any apoptosis, cell death paper, or
Yeah,
cancer paper. At least there is a Western blot or
of course
qPCR
they will do it, because
or a
again,
qPCR.
majority of the patients and majority of the cancer cells, they do
Mm-hmm.
have mutation in p53,
Yeah, yeah, yeah,
but
yeah.
exceptions are always there. Biology is full of exceptions.
Yeah, definitely. So I think so. Maybe we can take up this one last question.
Ah,
P53 gene mutation cause cancer in yeast as well as in like in mammalian cells. And
okay.
which type of cancer? I cannot
Okay.
understand.
No, I think I got the question, but the only answer to this is that we cannot say that p53 is causing cancer in yeast, right? Because
Yeah.
that is an entire cancer, has to be in some context, that normal cell and a cancerous cell, whereas in yeast, if we talk about, it is a single cell organism. So obviously technically we cannot call that yeast is suffering through a cancer. So I think that's the only answer I can give to this question.
Yeah, maybe
Yeah,
something more interesting towards this. If you have some minutes more, maybe
yeah,
five
sure, go
minutes.
ahead.
So something interesting, like have you tried to analyze the increased growth or something like that, focusing on this? So have you seen that? Okay, not exactly talking about cancer in East. That is very illogical, I would say. But something that makes it more proliferative, maybe. I don't know.
Yeah. So, Dhairya, that is a very good question that you have come up with. But if you must have noticed that we have worked with the wild type P53, which has a
Yeah, yeah.
normal role to play, right? So it is not going to give the proliferative benefit to the yeast cell.
Yeah, 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 yeah.
So obviously, if we could have gone ahead with the mutant P53s further, we
Uh-huh.
use mutant P53 just to show that whatever
Mm,
phenotype we are seeing is because of the
mm.
functional P53, not the other, just
Other.
the
Mm,
expression of foreign protein. So obviously,
mm,
we did not explore onto that aspect. But if
mm,
we could have made any other studies into that, we might
mm,
have got something. But to answer your
mm,
question, we
mm,
use only the wild type P53 in all our studies.
mm,
So it is
mm,
very unlikely that it will have some growth advantage to the cell
mm,
axis.
mm, mm. I see. I see. So we are having many questions, and many people are now started to come up with
Brainstorming
questions.
session, yeah. That is
Yes,
interesting. Yes, yes.
but we have time constraints as well.
Yeah.
We have to finish it completely. So maybe one last question.
Yeah, sure.
Maybe two last, I would say.
Okay,
uh,
okay.
one could be, so P53 is a tumor suppressor gene,
Hmm.
and its mutation will cause Crabtree effect, leading to
To
cancer type
cancer type.
condition. Like, does it lead to
Mm,
cancer type condition with this?
I am not
um,
sure. To
yeah.
be to be frank, I am not sure about this. Because again, as I
P-
mentioned, metabolism is a very complex process. Whatever mm, the pathways P53 is regulating, those are also very complex. Till
yeah,
date, nobody knows the exact interrelationship between those pathways, right? So I don't know.
it's
That's the
quite
only
complex.
answer I have. Yeah,
Yeah,
it
but like,
might
if
be, it
you
might not be.
but if do you have any idea uh, on interconnection? Maybe you can interconnection of mutation in P53 with Crabtree effect or something like that.
Mm,
uh,
See, that might be possible. If it is a mutant
mhm,
P53, obviously it will not have
maybe.
the regulatory effect of carbon metabolism,
Hmm,
right? And then it
hmm,
might lead to, let's say, what should I say? The cell might get converted into fermentative metabolic pathway. So yeah, that
hmm,
there might be some connection, but at this point, I exactly do not know the that key point where the 2 points can be connected. So that there is
hmm,
some missing link into it. But yeah, it would
hmm.
obviously have some effect to play.
Okay, so maybe this could be the last question of
Yeah.
the today's eukaryotic talk, I would say. So currently, 3 months are left for CSIR net.
Okay.
I
What
would say these are
strategy should we
most,
follow
yeah,
so that shall I read it out? Currently,
yeah, of
three
course.
months are left for
It's
CSR
all
net.
yours.
What strategy should we follow so that we can crack it easily? Okay, yeah. It would depend on person to person, but according to your experience, what advice will you give to current master and bachelor to crack such exams? Okay, so again, the only funda is you have to be constantly in the process. You have 3 months. I would suggest take out at least 1 hour from your daily routine and dedicate it towards preparation for the entrance exam. And as now you have very short period of time with you, what you can do is you can go to the previous papers of the exam and try to look for the questions and find the answer to it, because then you will get an idea. Okay, I'm reading, but that has to be directional study what you are doing. It just should
Hmm.
not be the gada mazuri type ka nahi chahiye. You should be, your effort should be directional, that should reciprocate into something. So when you have less time, the better strategy is what I think. You should go back to the the past question papers, 10, 12 years, whatever you can get, and try to look the questions, try to solve them. Then you will get an hang. Let's say for example, developmental biology, what sort of questions they have been asking. So if you have short span of time to study, you can focus only on that aspect, let's say initially. If time permits, you can study the complete topic, but at least try to study the important topics. I know that is very discouraging for others, but yes, you have to be strategic in that context.
Yeah, I would say, like when I was doing my thesis, I used to come back home at like sometimes 11 or 12, and still I used to keep this one routine. I have to come home, I have to read 1 hour. Uh, that's it. Whatsoever
Yeah.
I am very tired from the lab, I have to read 1 hour for
Correct,
the
correct. That's what. No,
entrance
yeah,
exam. And I
perfect.
did it for 6 months. I continued for 6 months, and in the last 15 days, I solved last 10 years paper.
Exactly. See, precisely that's what you
And
have
this
also done.
helped me. Yeah,
Correct.
and it helped me for gate. So maybe
Yeah, of
similar
course.
strategy could be for CSI or net.
True, true.
I would say. Yeah.
Yeah, yeah.
So maybe you can just tell it in very short. Maybe we can have a complete another session for this. I would say maybe you can keep up for that. But our participant is asking that uh, what struggles and problems you face during your
Ah,
research.
that
Major,
will need another.
I would I would just say one sentence. The major struggle that you face in East system is they don't grow. Every East biology should know
True,
this, that
true, true.
you set up a culture in galactose or lactose
Yeah,
or
of course, of
whatever
course.
system you are using, and
That's what you mentioned it very correctly. That if I can start talking about it, it is going to take another 4 to 5 hours,
yeah,
minimum four
exactly.
to five hours. But I'll just
Yes.
quickly share only one experience out of all the hassles I have gone through.
Hmm.
And the point I want to convey from that is that you have to be very cautious and observant. That's the main point that you should be imbibing as a scientist. So I'll tell you what happened was, obviously, as you might all of you now know by this time, that we worked with the galactose system. And obviously galactose is under glucose repression, the galactose regulon is under glucose repression. So any the minute traces of glucose, they're going to hamper the galactose metabolism, correct? So it was going fine. One day I came into the lab, I made the medium, the glycerol medium, and I induced the cells with galactose. And the strain, which should have given me the P53 onto the Western blot, it suddenly stopped showing me the bands onto the Western blot. Right? Antibody was same, the strain was same, everything was same. And then obviously the butter was like, Jaswandi, you might have done something wrong. So obviously, the guide, the first statement is this only. So then I have done, okay, I have taken out different glycerol stock of the strain. I tried that. I made all the buffers and everything fresh to Western blot. It did not work. I even used antibody from some other lab, considering that my anti-P53 antibody is not, it has gone bad, did not work out. And then ultimately, we use a reductionist approach, that eliminating one factor at a time, and it boiled down to the new dabba of the agar that we used from high media, and it had some traces of glucose. And that's why the and I think I have wasted almost 1 month in deciphering all that. So the system which was working so fine for me, I used to do Western blots 3 in a week or so, and suddenly it was like no band in the Western blot, it was all clear. And the only reason for that was we used the fresh dabba of agar, new batch of high media ka agar ka dabba, and it gave me trouble. So you never know what might go wrong into it.
Yeah, I mean, I usually used to do the same, and I still do it. If something stops working, I throw everything away.
Of
Whatever
course,
I
that you
use,
have to start afresh.
antibody,
Yes.
I throw my buffers, antibody, like diluted antibodies. It doesn't matter. It's costly, but I just throw
Of
them
course,
away.
yeah. Yes.
I don't want to risk anything. And I even make my fresh blot cuttings and everything fresh. Like I
Same.
take good Saturday, Sunday leave 2 days, and then from Monday I set up all the experiment. I thaw new cells. I do this like a whole new experiment. So
Right,
maybe
right.
especially this always works with the research. Something stops suddenly. Take a break
Yeah.
for yourself. Throw everything out. and start fresh.
Go watch a movie and
So,
then come back.
yeah, but it's good. You should need a passion for that,
Yeah,
certainly.
true.
So so thank you so much, Dr. Jaswandi Dandekar, for
Yes,
joining this kickstart session for our event. And I would say we got huge number of registrations and participants. So
yeah.
we had like more than 140 plus
Oh my
registrations
goodness.
this time. And
Yeah,
then since this is our first kickstart event for the SciComm Synapse, the
right.
new venture that we started recently, so it was like very good, I would say, and audience is ready to interact. So
Correct, correct.
I think
Yeah.
we will try to take this ahead.
Sure, sure, of
And
course. My best wishes are with you. And yes, once again, thank you very much for Ajinkya, first of all, and Dhairi also now. It was,
definitely,
very interesting interact with both of you. And obviously the entire team of the Psychom Synapse. And
yeah,
you have taken up a very good initiative. And of course, now I can tell you that in my capacity, I would be always available to you guys wherever you need anything from
definitely,
my
we would love to
side.
have you on boards or in team whenever you
Yes.
want to join. We are always
Yes,
open
sure,
for
sure. Of
that.
course, of course. Yeah, we can obviously think about that also, not an issue.
Definitely,
It was
definitely.
very
So
interesting for me also, to go back to my PhD days and, interact with the young minds here.
yeah, maybe someday we will talk about your how you handled research
Yeah.
and
And,
the
I do have some other story of my PhD work also, which I have not discussed. So you never
definitely
know.
that's always double sided in the
Yes,
research.
correct, correct. Because
So
you might be knowing that in our PhD, we usually work with not only one project, we have, backups
multiple
with us
projects.
ready
Yeah,
that if something doesn't work, we have another thing. So
yeah.
yeah, somebody asked me that what was the struggle. So the one of the struggle was that for initial 2 years, I worked with one project which
And
got
then
to
you,
some level, but then after that, it was, a stop. Okay, where should we go after this? And then whatever I have
yeah,
discussed today, I started work on that from my third year onward. So
definitely. This always happens. Like you hypothesize something, and
yeah,
then that's not the
exactly.
case ahead,
Yeah.
and now you have to completely shift your study. And I would say, in my case, it's happening from the first.
But it's good that if you face the problems in the beginning, it's better to handle those than facing problems at the end.
Hopefully,
Yeah,
yeah.
let's hope for the best and good luck for your both of you for your PhD.
Yeah, I'm just starting my PhD, so it will take like 4 years,
Arey,
and
just lage raho.
Ajinkya would be finishing. So
Yeah, he'll be finishing soon. Yes,
yeah, so Ajinkya is also connected to our community since we started, since four
correct. Yes,
years. We started with other name, and then now we have something which can be taken global ahead.
right,
And we
right.
have Soumya Mishra as well, who is also in our admins team. He looks upon
Okay.
the tech. He's a tech geek
Yeah,
guy, and
tech guru.
yeah, he's like a tech guru for us, and he manages all of our YouTube and everything. And we also had Surajit today, who is also in Max Planck Institute of Evolutionary Biology.
Oh, great.
And we have Pushti, who works with CES, and many more would also, maybe next time we would have uh, uh, more audience,
Yes.
definitely. So we have pasted the feedback link. Just let us know how we can improve our sessions next time, so that, yeah, like, this is our first session, so quite
No,
impromptu,
but
I
I
would.
would say it is a really good start. What you guys have organized, it's really commendable. For the first time, it's really commendable. Yeah.
I'll just take a minute, ma'am, to thank you
Yeah,
for your such an
Thank you, Ajinkya.
inspiring and insightful talk. It was really inspiring on a very realistic basis and
Yeah, thank
insightful
you.
on the basis of your PhD research, which you have done.
Thank you, thank you. My
Really,
pleasure.
really, really commendable talk. And of course, Dhairya, for such a wonderful hosting of the event.
Yeah,
You were
you
so
were very
interactive.
good. It was
Yeah,
fun
you
to
were so
interact with you.
interactive and yeah, very active with the whole talk. And Somilia on the technical side managed it so well with the YouTube and the Google Meet technical stuff. Yeah, and the audience, of course, for such a wonderful and
Yes.
keeping the
Thank
talk
you so much.
lively. That was really wonderful and being
And
so
thank you
patient.
for all the amazing questions from the audience.
Yeah, yeah. So I would like to thank everyone, and it was a very insightful and a wonderful talk.
Yeah.
Yes.
So I would
Thank
like to
you,
give
ma'am,
some glimpse
for
about
such
it.
taking out such time for us and
Yeah.
being our first speaker after we started with the new venture again with the new
Yes.
name.
Yeah.
And,
Yeah,
thank
Yeah. Thank you,
even
ma'am,
I
for
would like to
accepting.
just a second there. Yeah, yes, really thank you, ma'am, for such a insightful talk.
Thank you so much. My pleasure.
And on the all the audience on YouTube and G Media, I would like to request them then do like, share, and
And
subscribe
subscribe.
us.
yeah.
Thank you.
Yeah,
Yes.
please, audience, please do fill our feedback form. That will let us know how to improve on our
Yes.
events next time, and you can suggest
Yeah.
the changes which could be, benefit for both of us.
Yeah. Yeah. So I'll just give a short glimpse of, something that we would come up with soon. So we are coming up with some astrophysics and some exoplanet stuff next
Oh,
time.
nice.
So we'll come up with the SciChat event. So SciChat is something another, maybe something like this only. I think I would say more where we, interact with audience, and all of them they joined together and we discussed some aspects. Last time we had an introductory session where in the last part we also discussed about ROS and the metabolic pathways and stuff like that. And once we had quantum perception of biology, where we talked about quantum tunneling and
Ah,
stuff like that, which is related to biology, and how can we incarnate it in involve it in our biological sector.
correct.
So next time we would come up with like astrophysics, exoplanet stuff. So maybe people can
Interesting.
stay tuned. And then later we have some paranomical stuff as well, where we'll go towards exploring the science about in the darkness of paranormal investigations and stuff like that. So yeah, I would say people can stay up to date and will come up. So whatever love we get from audience, we would be enthusiastic more,
True.
definitely.
And if you're,
So
giving them plethora of topics like this, obviously you will
yeah,
get even more number of participants next time onwards.
yeah. Hopefully, we hope so next time.
Yes. Let's hope for the best. And the good
Yeah,
work always, have good energy to
yeah,
attract people. So yeah.
yeah. So we already
Once
passed.
again, good luck to all the
Thank
team.
you.
Thank
And
you, ma'am.
We
it
Thank
all
you.
was really fun to
yes.
interact with all of you.
Thank you. Thank you,
Hope
ma'am.
that today our India's team also plays so well
Yeah.
in
Yes.
the Asia Cup final. Yeah, we got century today. Hopefully, we have double century today on the
There.
fields.
Right.
Definitely. Okay, then. Thank you so
Okay,
much,
chalo.
everyone, for
Thank you.
joining.
Thank you
Thank you,
so
ma'am,
much,
for
everyone.
accepting.
Thank
Yeah. Chalo.
Yeah,
Bye bye, guys.
thank you. Bye, bye.
you. Bye. 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 Bye, ma'am.